Okay, so this is our third um, content review, and this is going to be content category 1C, which is mainly about genetic transmission. And so last time we talked about DNA, and this time we'll talk about how that actually affects us, So first things first, right? We talked a little bit about chromosomes last time. We talked about DNA packing and how it's stored in our cells, right? And so the major difference between prokaryotes and eukaryotes are the structure of the stored chromosomes. In prokaryotes, it's circular and it's not sequestered in a nucleus. It's just in this nucleoid space in the middle of the prokaryote. Whereas we saw with our mRNA, that our chromosomes and our genetic material is very much, has a strong boundary. Stuff can't get in or out to interact with our chromosomes unless very specific, signaling molecules are there. So already we have a big difference between um, the organization, right? And the other thing you'll notice is that here there is just circular like this, whereas here we have chromosomes, right? And we talked a little about the histones and the wrapping. So let me just show you to jog your memory the how chromosomes are condensed, right? Right here we can see we talked about how there are positively charged histones and with lysines right there. Positively charged histones are allowing these negatively charged DNA molecules to wrap around tightly and condense. So you can see the difference in prokaryotic and eukaryotic chromosomes. You have this long circular tangled up chromosome in a prokaryotic cell, whereas in eukaryotic cells, we have a bunch of linear chromosomes, not just one long big circular chain. Okay, so now we're going to dive in a little bit more into the specific structure of what we call a chromosome. Okay, so you have probably seen this kind of shape, this kind of X shape, and we're gonna dive into why this is it's a little confusing at first. We're gonna try to explain it well. So first things first, euchromatin is open chromatin, right? So we talked a little bit about the difference in packing. Euchromatin. Right here, yes. So heterochromatin is when they're very tightly bound, right? And we talked a little bit about methylation and acetylation and how they affect this, right? So euchromatin is open and available for transcription, and we can make RNA. Heterochromatin is not, right? And we can see here the chromosome, right? So how do both of these things happen? Basically, in the chromosome, we have this has two chromatids, and in the center, there's something called a centromere holding them together. And at the end, we have something called a telomere, which is just a repetitive, a bunch of GC nucleotides. And these are here because of something called the end replication problem. So this is jumping ahead just a little bit, but it's pretty cool, so I want to show you. So we had talked about DNA replication, right? And we talked about how the Okazaki fragments in the lagging strand are, much more complicated, right? Because of the directionality. What actually happens is every time you replicate your DNA, you lose a little bit at the end. And when this happens, because where is it? When DNA is being copied, only one of the two new strands is synthesized continuously, right? In the leading strand. Whereas the other strand is a lagging strand. And there are short regions at the end that can't be covered by a fragment. Because it can't be covered by the fragment, a little bit is missing now. You see at the end here. And when you have this little bit missing, oh, that could be a problem, right? We can't have missing DNA. Well, in this case, the safeguard against that is that the ends of the DNA are kind of junk information. They're just GC, GC, GC. And you can think of it, for some reason, somebody had told me, it's like the little thing at the end of your shoelace, that little plastic bit at the end of your shoelace, that it's like a cap that holds it together. But when that cap breaks, if that cap ever breaks, then the whole shoelace starts coming apart. So basically, that thing is there to prevent the DNA, to prevent this problem from reaching the actual important parts of the DNA. And this is also, you might have heard telomeres are something related to aging and stuff, right? Telomeres, let me see if I can find. This is a little bit outside I guess this is a little bit not super relevant, but telomeres are implicated in aging, right? Because the more this is a great picture, yeah. So the more you divide, the more the telomeres shorten, because every time you're dividing, you're losing a little bit at the end, right? And then when you have no more telomeres, you can't. The cells won't divide anymore. So there's a lot of research into, oh, well, can we extend our telomeres and keep growing or not age and stuff like that. So that's kind of an interesting thing at the end. Yeah, you see these GGC, these are the telomeres parts that are just repetitive, and they're non-coding. They don't code for anything, so if we lose them, it's okay. So back to our chromosome. Yeah, so chromosomes, karyotypes. Sure, okay, so karyotypes are how we stain and see our what's that word? It's not chromosome, how we see our genotype, I think, maybe? The word is slipping my mind. But karyotypes are very, very cool because I think it might be genotype. Karyotype. So karyotypes are basically, you can stain it in the lab and you can see your chromosomes. And you'll notice that they're numbered from largest to smallest, right? They keep going and going and going. And we have 23, we have two sets of 23 chromosomes, right? The one from our mom and one from our dad, right? And so in this case, this person is a woman because she has two X chromosomes. If it was a man, they would have one X and one Y chromosome. But all the other ones, you get one of each from both parents. And so now, where are we? So karyotypes, we just showed you a karyotype, right? You can stain and you can see your gene makeup, right? Now, the interesting thing is if you have a mistake, right? If you have a genetic error, you can see that. So this one, Down syndrome, also called trisomy 21, is where you can see that you actually have three of these, right? You're not supposed to have more than two. You're supposed to only have two. If you have three of these, this causes an issue, right? So we can see from genetic, we can see how our karyotype is impacted and how that leads to genetic issues. So we can talk about these abnormalities. Yeah, so aneuploidy is the specific word. When we mean ploidy, ploidy basically means the number. And aneuploidy means that there's an absence or a presence of an extra one, like something abnormal. And we can also have, rather than the numbers, we can have abnormalities in the structures of the chromosomes themselves, right? So if we look at, let's see. Yes, so there are various ways that we can have chromosomal mutations. So, and here they are. So if a part of a chromosome duplicates, Genome, yeah, that's the word. Why did I forget it? Yeah, thank you. Genome. So when we duplicate, you see how this just became longer, right? This part got duplicated. Inversions are when this part got flipped entirely. You can see it's yellow, white, but now it's white, yellow. Well, actually, wait, no, yeah, that's what it is, right? It got flipped around. Then a deletion is if a part of it just gets deleted straight up. An insertion is, like a splice, I guess, right? Like it gets put in here. And a translocation is a swap, essentially. So these are just various different ways that chromosomes can be mutated. And we're going to see how that's actually happening. What's going on to have chromosomal mutations happening. But basically, these are the various, like 5 different major types of chromosome mutations. Now, there's also in the DNA, we can have mutations. And I think we touched on this last time a little bit. Mutation. Yes. Oh, okay. So there's a lot to go over here. Okay, cool. So mutations are when, we have our DNA and we need to copy it correctly. We need to transcribe it correctly. We need to translate it correctly, right? At any point, we can have errors, right? And we try our best to not have those errors. But as environmental factors can increase your chance of having mutations. If you're a smoker, if you have carcinogenic exposure, right? These things are, or even stress, right? Things that harm the integrity of your biological processes will increase the chance of your mutations. So what kind of mutations can you have? Well, you can have, the first group is called point mutations. So actually, let me see if I can. Okay, the first group is called point mutations. And so we can have 3 different kinds of mutations that are point. So a point mutation just means that, like, for example, if we were dealing with like ATCG, right? Then when we copied it, we accidentally turned it into AUCG or something, right? So a point mutation is basically we made a mistake in one nucleotide. So now, how does this matter? So for example, if we have, we know about codons, right? We know each of these things will give us three nucleotides at a time will correspond to an amino acid. A silent mutation is one where the mutation doesn't actually affect anything. So for example, we talked about the third base wobble. The third base wobble means that CUU and CUC probably both code for the same thing. I'm not 100% because nobody memorizes that, but it's pretty likely that they, but I'll just check right now. Oh yeah, actually that's the exact example. CUC and CUU, right? So they both do the exact same thing. So where am I? Yeah. So in this case, this is what we would call a silent mutation. If we turn the CUU into a CUC, it doesn't have any effect on the protein that gets created. So therefore, this is a silent mutation. We didn't change anything. Then we have, one second. So then we have missense mutations. So missense mutations are when if we make, so for example, let's say instead of this, it was, instead of AUG, CUU, and then CCU, this might have a big effect, or guaranteed this will have a big effect. Instead of CUU, it was GUU. This will have a big effect for sure, because this is going to change the amino acid that we get. Because the CUU will code for one amino acid, the GU will code for a different amino acid. That's called a missense mutation, because we've changed one amino acid that was in our chain. Now, a nonsense mutation is when we screw up the whole thing. And that would be, I think, so for example, UA, right? If we accidentally turn this into UAA, now UAA is a stop codon. So we're going to stop translation. And so that is going to screw up our whole protein. Now we have a non-functional protein. So these are the three types of point mutations. Why point? Because we're only changing one nucleotide, only one at a time. So that's how point mutations work. So let me just show you the definitions here. Okay, yeah. So point mutations, different codon that codes for the same amino acid, different codon that codes for a different amino acid, nonsense, different codon that codes for a stop codon, right? So you can see why it's a silent because it doesn't do anything. A missense, okay, it makes a small mistake. Nonsense, we screwed up the whole protein, right? So those are point mutations. Then frame shift mutations are a little bit trickier. Frame shift mutations kind of screw up the whole thing in a way. So basically, if we had AUG, UUA, and then GUU or something, and by some horrible error, if A got deleted entirely, then we end up with AUG, UUG, UU, and then whatever is like next in the chain. If this is like CAC, then we end up with C here, and then we end up with AC here, and then whatever is next in the chain. So you see how everything got shifted by one. So this is having a huge effect. This is screwing up every single amino acid down the line, right? Yeah, amino acid down the line. So basically, that what I just showed you is a deletion, right? We deleted the A here. But you can have the opposite effect. You can also have an insertion. So if you have AUG, UU, and now let's say we insert a C, then this is going to get pushed down, AGU, and then UCA, and so on and so forth, right? So these are two different types of, or rather the two major categories of polymerase errors, point mutations and frame shift mutations. So now, yeah, and we talked a little bit about the kinds of damage that can be causing these mutations, right? If you have reactive oxygen species, if you have a high oxidation like peroxidase and stuff, these are very they have radicals, right, on their oxygen, so they mess around with everything. That's why you'll often hear about how healthy antioxidants are, right? Because we're trying to get rid of these reactive species that can cause DNA damage. Cross-link bases are when DNA bases accidentally get covalently bonded, and that's just not supposed to happen, right? They're only supposed to be H bonded to each other, not covalently bonded. So that's a whole mess. Physical damage could also occur. I think that would be more along the lines of I'm not sure actually what kind of physical damage, what the situation would be for physical damage in a cell. I'm thinking more along the lines of exogenous damage like radiation, X-rays, and there's also poisoning, right? There's also things that explicitly go in and screw up your DNA. So those are the kinds of things that can cause these mutations. Oh, actually, wait, we have a picture right here explaining everything with see, this is a silent mutation, this is a missense mutation, nonsense mutation, codes for a stop, and a frame shift mutation. Yeah, everything is here. Okie dokes. Oh, one other thing here is transition mutations, where so a transition mutation is a purine got swapped for a purine. A transversion mutation is a purine got swapped for a pyrimidine or vice versa. So here's a question. What do you think would be worse? Do you think a transition mutation from A to G or a transversion mutation from A to C would be worse? So how can we think about what would be good, what would be worse, and what would be better, right? So if we're talking about the structure of the DNA, right? If the DNA is expecting an adenine, right? Which would be worse to show up, a cytosine or a guanine?
Cytosine,
A cytosine, right? And why do you say a cytosine?
because it's structured differently.
It's
And I'm
yeah. Sorry, go
sorry,
ahead.
I was just gonna say, when it's being paired with another base, it might it changes like the amount of hydrogen bonds. Would that be part of also the reason why?
Yeah, absolutely. Because we're talking about the bonding, right, between the two DNA strands, right? So if we have if we're supposed to have an adenine, but something shows up that has totally different hydrogen bonding, that's a problem, right? That's a problem for our DNA bonding. So structurally, this is a little bit more similar, so maybe we can sustain it. But when we have a swap to a totally different category of nucleotide, right? That can pose more of a problem. So that's just an interesting way to look at it. Remember, we talked about how structure relates to function and vice versa. If you take one thing away from all of biology, that's ultimately the final lesson. So good. So any questions on mutations in DNA in general? Oh, we also have this thing called thymine dimers, which it caused by UV light. That's why you're supposed to put on your sunscreen and not get too much direct sunlight, because you can have DNA damage, because this will cause a covalent bond between your DNA, and then that's not good. So yeah. Okay. So now we can talk a little bit. Now we're going to get into the good stuff. The good stuff. So let me just clear this off. Okay. All right. So okay, so the normal way that we think about genes, or the way that we were taught about genes, is with Mendel, right? And like the flower plants and stuff, right? So it's a little bit more complicated than that, because in humans, right? In smaller life forms like that tends to be, oh yeah, like this one thing connects to this one other thing. But in humans, right, it's not that simple. Otherwise, we would just, find the specific gene that turns on cancer, and we would just turn it off, right? And then easy peasy, we solve cancer. Obviously, it's not that simple. In our genes, we have something called, we have a single gene is encoded by a lot of different, or rather, a single trait has a lot of contribution from a lot of different genes. So it's not as easy to just go and be like, oh, flip the switch here. No, that switch is connected to a bunch of other switches, and we don't necessarily know. We haven't explored that fully yet. So normally we have 23 pairs of chromosomes in each cell, right? And that gives us 46 chromosomes, because we have 23 pairs. So each pair has the same chromosome we saw in that karyotype, right? Each pair, this is a pair of chromosomes, and we have 23 pairs, and we have 46 chromosomes in total. Yes. So where are we? And we are diploid because we have two sets of 23, right? And our ploidy is the number of sets of chromosomes that we have in the cell. So we have two sets, so we're diploid. Now, the sex chromosomes, right, as we saw, are a little bit different. And these are just to determine our sex, right? We know that females are XX, males are XY, and the other 46 chromosomes are autosomes, labeled from 1 to 22, and they are identical, or at least they should be homologous, right? We're going to talk about what homologous means. So Alleles. We have to talk about alleles now. Alleles are gene variants, right? And we know, you'll probably remember, like when we did the pea plant stuff, if there's a Punnett square here, right? These are alleles, the big A and the small a, right? Like the same gene, but with different traits, right? So that's what we mean by an allele. And when we're looking at this kind of chromosome here, right? So let me find a picture of a locus. Yes. Okay, this is a good picture. So this is a homologous pair of chromosomes. Homologous meaning that they are the similar length and they're coding for the same genes, right? Everything in this chromosome. However, just because they're coding for the same gene, aka in this case, the flower color gene, it doesn't mean that they are coding for the exact same trait in that gene. So, or allele in that gene. So they're both in this region, or aka locus, in this position, in this locus of the chromosome, they are both coding for a flower color. However, the left one is coding for white, and the right one is coding for purple. I don't know why they chose totally, not matching colors, but basically the point is that the left one codes for the flower color gene and it codes for a white flower. The right one codes for the flower color gene, but it codes for a purple flower. So these are homologous chromosomes, and this is where our genetic variability comes from. So we're going to explore this. So yes, while all alleles for a gene are found at the same position or locus, variations in gene sequences confer different characteristics for gene activity. And it's this complex interplay between the genotypes that leads to the physical characteristics, aka phenotypes, right? So yeah, each human cell carries at least 2 alleles of each gene, right? One from the mom set and one from the dad set of chromosomes. Okay. So now, before we can talk about Mendelian genetics, we need to do a very brief overview of mitosis and meiosis. Yeah, we should probably do that. Any questions before we dive into any questions about like the start of like chromosomes specifically right now, before we start talking about mitosis and meiosis? Okay, and let me just see if there's anything else I want to talk about with chromosomes. No, I think we're just going to get right into mitosis and meiosis then. Okay. So we're going to do, so mitosis is actually covered again in concept 2. So we're not going to do a very deep dive. We're just going to briefly explain it so then we can talk about the genetic implications. So I think we've all probably committed to memory prophase, metaphase, anaphase, telophase, blah, 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 right? Basically, and we will talk about the cell cycle next time, but basically, mitosis is how our cells duplicate, right? We've seen how our DNA duplicates now. This is how our cells duplicate. And mitosis basically involves, let me see if I can get a better picture. No. Oops. Where am I? Okay, here we are. Oh, right here. Yeah. Okay. So prophase is the first step, right? Where our chromosomes get condensed into this form, into this, what is it called? It has a specific name. No, it's just chromosome. Oh, right. Okay, yes. Okay. This is so annoying, and it took me the longest time to figure it out, right? We see here that a chromosome is, chilling on its own, right? If we just look at, so like right here, look, a chromosome is just chilling on its own right here, right? And then there's another chromosome here, right? If when the chromosomes are duplicated, they look like this. They look like X's. Do you see the difference, right? They look like an X here, but this X is still one chromosome. This is very important and it will screw you up. So try to follow along with this and ask me if you don't understand. A chromosome, let me try to draw it so I can be very clear. Let's imagine we're talking about 1 right now. We're talking about 1, right? A regular human with unduplicated chromosomes has a chromosome 1 set that looks like this. These are their chromosomes. They have 2 number 1 chromosomes, one from mommy and one from dad, right? Now, if we begin, and also the number of chromosomes that they have here now is 2n, right? Because here's, and this is 1 set, and this is set 2. So then they have 2n, right? Now, if they duplicate their chromosomes, so we are getting ready to duplicate the cells, right? To duplicate the cells, we need to duplicate our DNA first, right? So that the new cell can get a set of DNA, right? So we need to duplicate our DNA. When we duplicate our DNA, we have to make a second version of the chromosome, like this. However, sorry, not chromosome, chromatids, because this thing here, this X, is still called one chromosome. This is very, very annoying for me anyway, but this is still one chromosome. It's a duplicated chromosome that has doubled the number of chromatids. So in this case, we had, you can say 2 chromatids, but now we have 4 chromatids. Okay, so that basically, just be aware that once we duplicate the chromosome, it doesn't mean that we suddenly have 96 chromosomes. We don't have that. We always have 46 chromosomes. However, once this gets split, then there will be a total of 96 chromosomes across 2 different cells, and each cell will have their own 46 set. Any questions about this? This is very, very important. Does it make sense?
So just to clarify, the sister chromatids are only like during the DNA replication of like mitosis and
meiosis, yes. The sister,
meiosis.
exactly,
Okay.
yeah. So normally they wouldn't be looking like this. Normally they would be looking like that. Okay, so that's good. Okay. So now we want to talk about, yes. So that's why. So in this diagram here, there's only 2 chromosomes. Then in metaphase is when they line up in the middle, right? And they get pulled apart in anaphase. And then now we see that we have 1, 2, 3, 4 chromosomes, and each cell still has 2. Notice how the first cell has 2 chromosomes, and the last 2 cells each have 2 chromosomes. So the cell always has 2 chromosomes. That's how the math works. So this is just mitosis. This is just for us to get to the point that we need to talk about with meiosis. Okay, so meiosis is different, and meiosis involves us creating. In mitosis, we start with a 2n and we end with a 2n. So what do I mean by that? This is a 2n because we have 2 times 23 chromosomes, essentially. So we have 46 chromosomes. And that's what 2n means. Here we also have 2 times 23 chromosomes. But in each new cell that we just made. Because you can see if we so going by just the diagram, right? If we go by just the diagram, we have 2 times 2. By the diagram, there's two sets. There's one. Oh no, they're actually just doing one here. They're doing two sets of one. This is 2n. This is 2, 1. So this is a 2, 1, and then this is a 2, 1. And then this is a 2, 1. So now if we go to if we go to meiosis, we can see how the numbers are different. Now we start with the same situation, but when we go to metaphase, the way that we lined up here in the middle is not the way that we line up here. Now the homologous chromosome pairs line up next to each other because their goal is to get split apart. In this case, their goal is not to get, the goal here is to have a uniform distribution of all the chromosomes. It's the opposite here. We don't want a uniform distribution of the chromosomes. We want it to be distributed in a unique way. So then we split this apart, and now we have a chromosome, they're both the same chromosome, but with different alleles, right? This one is red because it might code for like the red color, whereas the green one codes for the green color or whatever, right? This is an oversimplification, but that's what I'm just as an example, right? So then this one and this one are going to get split further. And then this phase is the one that looks like mitosis, because notice how the chromosome goes to the middle and gets split in the middle, just like we did mitosis, where it comes to the middle and gets split in the middle. And so now the important thing to notice here is that first we started our chromosome count is 2, 1, right? 2 sets of 1, right? Or 2n, where n is the number of chromosomes, right? So it could be anything. If there were 4 X's here, then this would be a 2, 2, because it would be 2 sets of 2 chromosomes. So this is 2n, right? Here, when we get to just this X, this is still 2 of 1. But then when we get to the last part, that 2 of 1 is getting split into 1 of 1s. So the whole point of meiosis, the whole big difference between meiosis and mitosis is that in mitosis, we go from 2n and we maintain 2n. In meiosis, we go from 2n to 1n because we split the after we made 2, we split them again a second time. So, okay, what's the point of all this? The point of all this is how we get genetic diversity. So let's go back to our genetics. Right here. So here we have our 2n getting split like this, and then getting split further. And then we have only one chromosome in each of the gametes that we made. And there are some rules here discovered by Mendel, who probably everybody knows. And Mendel was pretty brilliant because he figured out these things before we even understood like chromosomes and mitosis and stuff. Like he just figured them out based on observing the patterns. but that he saw in the plants. So what he figured out was that the law of segregation, meaning that these are getting distributed independently. They're not staying linked. They're getting cut in half and then cutting in half again. And so gametes can be given out independently and chromosomes can be distributed independently. So alleles are equally distributed to daughter gametes from a dividing mother cell, and the two allelic copies of genes in the diploid organism are equally segregated into the gametes, such that the two gametes that are formed both receive one of the pairs of alleles. So that's the law of segregation. The law of independent assortment is that it's this part. Hold on, I have a better picture for this. Yeah, okay, here. So yes. So when this is metaphase 1, where the homologous chromosome pairs are lined up in the middle and then getting separated into two cells, and those cells will get separated into 4 cells. Now, the thing is that these guys can line up randomly, right? Here we have a blue and the red and a blue and a red. But what if it was blue, red, red, blue, right? This is totally fine. We're not breaking any rules here. This is the correct homologous chromosome pair, and the right ones are connected to the right ones. So there's nothing wrong with this. And this increases our genetic diversity because once we get the two, right? In this case, we have totally blue and totally red, but here we have blue and red and blue. And so once this splits down into the gametes, we're going to get different allele combinations, right? Here we would get A, capital B, and only lowercase a, lowercase b, right? But here we get capital A, lowercase b, and we get lowercase a and capital B. So this is the second, so we have two of Mendel's laws, the law of segregation and the law of independent assortment. Okay. And now we get to talk about genetics and dominance, and I'm sure this is a, yeah, everybody studied this for sure, right? Like this has to be part of bio or bio 2. So yes, so, and I really like this because I really like Punnett squares. It's fun to me. So we have genotypes and phenotypes. And so let's talk about the classic pattern. And this will go a little bit further than probably what you studied, unless you took genetics. If you only took a regular bio class, then you will see a little bit more than you probably did before. What's it called? Is it Punnett? What did I call it? Dihybrid? Oh, okay. So maybe around here somewhere. Okay. Anywho, the Punnett squares, right, are a way for us to see to predict the genotypes that we can get based on the parents' genotypes, right? And so this is a monohybrid cross, which is simply the parents. You cross both parents, and you can see the potential offsprings that the child can be. So, and of course, it's a 1 in 4 chance to get a particular genotype. However, this doesn't mean that you will see the same thing with the phenotypes, right? Because of the fact of dominance and recessive. So I had a good one here, but I don't know where it is. No. Well, anyway, there's basically a ratio of, yeah, okay, here. Okay. So basically, if you have, does everybody know how to do Punnett squares? Or do we need a refresher? A refresher is fine. Okay, let's do a refresher on Punnett squares. So we have just talked about alleles, right? Alleles are these genes that we're talking about, and in this case, we are labeling them as capital or lowercase. So, for example, in this case, they're saying that a capital W is going to give us a blue flower, whereas a lowercase w will give us a white flower. And if it's a capital letter, that means it's dominant, right? So when we have two alleles, and if we have two, like, so a kid, so like an offspring can have this WW, right? And so what will they be? Will they be blue or will they be white, right? Well, if the capital one is the dominant one, so it overtakes the white, so then it will overall be blue. Then if it's, so the simpler ones, right? If it's WW, then it can only be blue. If it's WW, it can only be white. And if it's this, it will be blue. Okay? So from these rules, we can establish like predictions of what will happen to our offspring. So we can see that if it's a dominant, and this is called homozygous. Homozygous means you have the identical for the dominant, and homozygous recessive is you have the identical alleles for the recessive trait. So if a homozygous dominant and a homozygous recessive both cross, you will always get blue flowers. You'll always get blue, but they are heterozygous because their homozygous blue would be WW capital. Heterozygous blue is one capital and one lowercase. So this is always the case, but don't worry about like, oh, I have to memorize that homozygous dominant and homozygous recessive gives me heterozygous dominant. You don't have to do that. You can simply draw out the Punnett square and see what you get because the Punnett square works. So, whoops. Oh, I didn't want to delete that. So now we have a heterozygous crossed with a, wait, is this heterozygous? This is not heterozygous. Oh, they just made a mistake here. This should be a lowercase w. They fixed it here. So a heterozygous, a capital W and a lowercase w. Let's just fix that a little bit. A capital W and a lowercase w with a homozygous recessive, right? And so if we draw this out, right, we have a capital W here crossed with a lowercase w here, and that gives us this, right? And that gives us this, and that gives us this, and that gives us this. So we get 50% are heterozygous and they're blue, and 50% are homozygous. So that this allows us to see, this allows us to predict, you know, what we'll see in the offspring generation. Now, one thing I wanted to show you guys is that this thing, this is a dihybrid cross where you do the where you have to account for two traits. in both in across both parents, right? And you can see what will happen. You can get a lot of different options here. And the other thing that, where is it? I want to account for phenotypes and genotypes. Phenotype. That's very surprising that they didn't talk about it here. Let me see if I can find it somewhere else because it is important. Maybe here, phenotype and genotype. Yes, this is what I was looking for. So we see here a cross between a heterozygous dominant and heterozygous dominant. When you do that, you get 3 purples and 1 white, but the 3 purples are not the same. One of the purples is a homozygous dominant purple, while the other 2 are heterozygous dominant purples. So this makes a difference in the ratios that we see. The ratio phenotypically is 3-to-1 for purple, but it is not 3-to-1 for the genotype. The genotype is 1 to 2 to 1 because of this BB, capital BB. This is not the same thing as the other 2, even though phenotypically it looks like it's the same. So that's an important thing. And yeah, so the ratio, important to know. Talk about that. Okay, so now we can talk about a little bit about, we talked about recessiveness. Now we can talk about, so I just told you that, you know, if you have a capital W and a lowercase w, then the capital W wins, right? And the flower will be blue, right? This is not always the case. This is specific, this is how we were taught, right? But specifically, this is called something called complete dominance. Complete dominance means that one allele is totally masking the effect of the recessive allele. But that's not always the case. There's other things called, for example, codominance. So codominance is when, if you have 2 different, you can think of it as 2 different capital letters, right? And the most easy thing to see is the AB blood type, right? Because that's 2 capital letters. That's not necessarily, but like you can just think of it as that way. Not only is A being expressed, but B is also being expressed at the same time. So this is codominance. They're both winning. Nobody is, you know, masking the other. So this is what we call codominance. And then we also have incomplete dominance. So incomplete dominance is a blend. So I usually think of incomplete as in between, right? It's an in between. So if you cross a red with a white and you get a pink, right? Did the red win? No, because this isn't red. Did the white win? No, because this isn't white. You got a totally new different thing. That is a in between of the 2 beginning of the 2 genotypes that you started with. So this is incomplete dominance. So it's not always that, you know, the bigger letter wins. There are different variations in how they win and how it's expressed. Yeah. So that is the basics of genetics, right? And now we're going to get into some of the interesting stuff that you might not have seen if you haven't taken a genetics course. Some of the little bit more deeper stuff. So any questions about, you know, the basics of genetics and the basics of genotypes and phenotypes and all that stuff? Okay, so far so good. Okay, good. If you have any questions, you can drop it in the chat and I will get to it and we can get started. So yeah, okay. Yeah, so now we got to talk about, so basically we've just covered the main, right? The Mendelian genetics. Now we have to talk about all the exceptions, all the interesting, weird stuff, right? So what we've talked about here is autosomal genes, right? There's also sex-linked genes. And the sex-linked genes are a whole different topic, right? Let me see. Sex-linked traits. So males express recessive X-linked traits more than females. Why is that? Why would males express recessive X-linked traits more than females? Well, a male is an XY genotype and a female is an XX genotype. And let's say that this X, right? Let's say that the female, so let's say that a female has an XX thing, but one of her X's has a recessive trait, right? She herself is not going to show that because she has an X that has dominant. Can you explain how some genes are passed and some are not? Somatic. Somatic? Somatic recombination? That's the only thing I remember with somatic.
I was saying that I'm not sure. Like some things are like within the cell line, but then some things are not. Like it's like some cancers are not genetic, I guess, so they can be passed on. But I'm forgetting the exact word for that.
That some things are not passed on?
Yeah, some genes are not passed on, but they're within like one person's body.
Okay, well, I mean, there's a lot of points that could be referring to. One, I think one of the major things you could be talking about is how, we only get a specific gamete from our mom and from our dad. And you know, the actual genes that we're getting were selected randomly from the when we did the meiosis, right? So if we started with a full thing, we're only getting one of these four. So we can't get every, but I'm not sure if that's what you're referring to. You're saying genes that don't get passed on.
Like somatic mutations are not like transmitted to the offspring, but like other things can be. Like other types of mutations can be, transmitted and passed on to the offspring.
I understand what you're saying. Mutations that get passed on to the offspring have to be in the gametes. If it's not happening in the gametes, then they wouldn't get passed on to the offspring. So if it's a genetic mutation that can be passed on through fertilization, then a child will get it. But if it's something that happened, like let's say, okay, this is a good example. Let's say I have, well, I don't know why I would say I. Let's say a random person gets radiation poisoning and they have mutated DNA in their arm, or let's say there's somebody who doesn't put on sunscreen, so they go outside all the time, and then their arms get skin cancer, right? They have mutation in their DNA in the skin because they're constantly exposed to UV light. If this person has a kid, that kid is not going to suddenly have skin cancer because the UV light was not affecting his gametes. They were affecting his somatic. That's what I understand what you mean by somatic now. They were affecting the parent's cells, but those skin cells don't have a connection to the chromosomes that we will pass on to our kids. That has to be gamete affected. So a mutation, in order for a mutation to be passed on to a child, it must affect the gametes. It must affect the chromosomes that we pass on to our kids specifically. Okay, I feel like I remember seeing a question about this somewhere, but it's been so long. But you just suddenly like reminded me about all that. Okay, yeah, good question. Now what was I talking about before? Hmm, what was I talking about before? Hmm. Oh yeah. Sex link. Okay. Yeah, I remember where I was. Okay, so yes, sex link gene. Thank you. So, and I just saw this random card while I was looking at that. This is kind of cool. Recombination. Is this when did we talk about recombination? I think back with I think probably when we talk about meiosis in more detail. Yeah, okay. So we don't need to cover it right now. So let's talk about sex link. Yes. Yes. Okay. So we were talking about a parent, right? A mom who has an X and an XI, right? XI being the affected whatever, right? Some kind of recessive trait. And so now if she has a son, right? A son can be an XY or he can be an XIY, right? But if she has a daughter, that daughter can be X, assuming that the father is like a normal XY, right? The daughter can be XXI or just an XX, right? Because the mother may pass on her X or her XI, and the father will always pass on a normal X, right? So this is what the daughter can get. Whereas the son can either get I don't know how that happened. The son can either get, you know, uh, oh no, this got screwed up. There should be, the son can either get a normal X from his mom or the recessive X from his mom. So now notice here, is the sister ever affected by the recessive gene? She's not, because she has this X all the time. She always gets a normal X, at least one. So if she has a normal X, that one, if it's dominant, it's masking the recessive trait. It is not the case for the son. The son does not get to have a normal X that can mask the recessive X. So if he gets the recessive X from his mom, then he will show this phenotype. He will have this trait. That is why we're saying that males express recessive X-linked traits more than females, whereas autosomal traits are expressed equally by males and females. Why? Because we don't have this Y chromosome factor when it comes to autosomal traits. How does the autosomal trait compare? An autosomal trait would be something like AA and AA, right? You see here, there is no influence of a Y chromosome here. It is equal. It could be the mom, it could be the dad. The mom could be affected, the dad could be affected, right? A son could be affected, a daughter could be affected because there is no influence of the Y chromosome. So let's talk a little bit more in depth about this because this is very, very commonly tested. They love asking about this. Okay, so, okay. So the X chromosome is much larger than the Y chromosome. Yep. Contains more genes. Such genes follow an X-linked inheritance pattern. Yes. Right. So here, color blindness is a common one. Yes. So we have a carrier female. A carrier means that they have the recessive trait, but they don't express the trait phenotypically, meaning that this mother has the gene, but she is not colorblind because she has a dominant X that gives her color vision. Now, if she, um, and also, right, the only, actually, let me not jump ahead. Let's finish this first. If she has kids with a normal male, normal meaning not colorblind, then she will have a normal female or a carrier female who is again not colorblind, or a normal male or a colorblind male, right? So just to be clear, all of these people have color vision. Only the colorblind male is colorblind. So then the question is, how could we get a colorblind female? Is it
The
even
dad
possible?
carries it.
The dad has
Oh,
to carry it, right? Yeah, yeah. No, you're right. I'm just asking. The dad, if the dad is a colorblind, right? Will the daughter always be colorblind?
no.
No, right? Because if we turn this into XC, right? And we can see that this will be XC, and then this will be XC, right? And then this will be, oh no, it's a carrier female, so that's correct. It's a carrier female, and then this is that, yes. So we can see that this will now be a carrier female, and we can get a colorblind girl like this. So sex and traits are a little bit different. Yeah, a little bit different than our autosomal traits. But this is something that is really important, and they talk about it a lot. Yeah. So, and the crux of this is because sons have to get the Y chromosome, right? That's the main reason that everything is getting changed. So as long as you keep that in mind and you can draw your Punnett square, then you'll be good. So we talked about codominance. We talked about codominance and incomplete dominance, right? Yeah, we talked about AB blood type and we talked about pink. Yeah. So we did that. Okay, so now we get into a little bit more of the nitty-gritty. So now we introduce this idea of penetrance and expressivity. And I think it's best to look at a picture. Yeah, okay. So penetrance is the proportion of genotype carriers that actually express the phenotype. So notice how before we started super simple and super basic. If you have capital A, lowercase a, that means that you express the dominant color, right? Now we're throwing in more variability because genetics is not simple, right? We can tell from our everyday lives genetics is very complicated. How is it complicated? Because there's all these small little variations and parameters and modifications and stuff, right? So now we're introducing the idea, okay, hold on. What if you have capital A and lowercase a, but does every flower that has capital A and lowercase a actually turn out white or actually turn out blue or whatever the dominant color was supposed to be, right? That is the idea of penetrance, that how many people actually express the phenotype once they have the genotype, okay? And of course, you have two different extremes. If you have complete penetrance, it's, yes, it is that simple. If you have it, you show it. If you have the genotype, you show the phenotype. If it's incomplete, it's like, well, actually, it's not that simple. You might have the genotype, but not everybody with the genotype actually shows that phenotype. Okay, and so an example here with penetrance is right here, right? In this top row. Every person with this genotype is showing that blue color. So we see like 100% of people that this gene or this allele has high complete penetrance. Now, if the box on the right were, if the box on the right here were white, then this would now be incomplete penetrance because we have an example of that genotype without the phenotype. So that is penetrance. Then we have expressivity. Expressivity is the variation in phenotypic expression when an allele is penetrating. So in the second row here, we see complete penetrance means that everybody is showing blue. Everybody has the phenotype, but not the same shade, right? Not the same shade. The expression is slightly different. There's varying levels of showing the it's like two steps. It's like, yes, I have the genotype. Okay, yes, I am showing something because of my genotype, but am I showing the exact same thing as everybody else? No. So therefore, there's variable expressivity here. If it was this first one is complete penetrance and constant expressivity. I can just write that in. And so this is complete penetrance. Everybody with DD shows blue, and they constantly showed the exact same shade of blue. So this is complete penetrance with constant expressivity. Now the second one, as we can see, is complete penetrance. Everybody has DD, everybody shows blue, but a variable shade of blue. So variable expressivity. And then we get to incomplete penetrance. Not everybody's showing blue, right? Some of them are not showing the blue, even though they have the DD. And then lastly, we have incomplete penetrance and variable expressivity, because it's not only are some people not showing the blue, but the people who are showing blue aren't always showing the same shade of blue. So this is a whole bunch of, modifiers and up and downs, but don't get confused through like all the different combinations. Just understand them on their own individually. Penetrance, if you have the genotype, you show the phenotype. Expressivity, if you show the phenotype, do you show it the same exact way or a little bit differently? Those are the two things that you need to know. And then all the combinations, you can just mix and match. Any questions about penetrance and expressivity? Okay.
Sorry. So for penetrance, is it just like you would still have the gene, like the expressivity is like it can show different like levels, like it can be expressed in different ways or not be expressed. Like I still get those two confused.
So everybody has the same genotype here. Penetrance and expressivity, we only talk about, so if we break it down into steps, step one, or question one, do you have the genotype, right? If you have the genotype, now we can start talking about, analyzing the penetrance and expressivity, right? So if yes, then we go to next two, right? Yes, then we go to two. Now, do you express the phenotype? If the answer to this is yes, if it's always yes, that means that it's complete penetrance. If it is not always yes, then that's incomplete penetrance. Does this part make sense so far?
Yeah.
Okay. Now we get to, do you express the phenotype the same way always? If it's yes, always the same, then this is called constant expressivity. No, not always the same, variable expressivity.
What do you mean the same way every time? It has to
So
be like purple or white every time?
no, so we're not talking, so purple and white is kind of talked about in question 2. Although, okay, so we're not actually talking about purple and white now. We're only talking about purple. because purple and white would be two different genotypes. Purple would be like, I don't know, I'll just use A, or I'll use P. Purple would be PP or big P, little p, right? Whereas white would be equals purple. And then white would be this, right?
Mhm.
What we're talking about here is not between these two, but just talking about a single genotype. So then the question would be, do you have this genotype? Yes. Therefore, do you express the purple phenotype? If it's always expressing the purple phenotype, then it's complete penetrance. But if you're not always expressing the purple phenotype, if for some reason you just don't show the purple phenotype for no reason, right, then it would be incomplete penetrance. And when I say that you don't show the purple phenotype, I'm not saying that it's white. I'm saying that there's just an absence, right?
Mhm.
Maybe this thing is like by default black. So I'm saying that, if you have the genotype, if you have a specific genotype, are you always expressing that specific I think I might have switched it up. If you have the genotype, do you express that phenotype? If you always express that phenotype, then this phenotype, the big P, has complete penetrance. If not, then it has incomplete penetrance. Then, do you always express the same shade of purple? That's how we could talk about expressivity in the PP genotype.
Mhm.
And then if it was always the same, then it's constant expressivity. If it's not always the same, it's variable expressivity.
Okay, this makes
Is it
sense.
good?
Thank you. Yep.
Okay, good. Okay, good. All right. Yeah, good question. This I had to clarify that we were talking only about one genotype here. Okay. Okay, I put it in the chat if you need it. So now let's go to our next little topic. Oh, they used broad and narrow instead. Interesting. That's fine. Yeah. So now let's talk about genetic linkaging. Yes, okay. So in the previous discussion, it was stated that sets of genes located on different places on a chromosome would assort independently during meiosis. Yes. This holds largely true for alleles of different genes on the same chromosome. Genes located far apart are rarely inherited together because of genetic recombination during meiosis. So genetic recombination is this. Yes. Genetic recombination. Okay, so we talked about, in meiosis, can I find a nice picture of meiosis? In meiosis, right here, these guys are hooked up together, right? But they're actually interacting with each other. And the way that they're interacting is something called the synaptonemal complex. Let me see if I can find it. Synaptonemal complex. Yes, here we go. Yes, okay, now we're here. Okay. So now we can talk about this. So once we get to, oh yeah, right here. Crossing over during prophase 1. So we have our, these guys are made here. And then they're hooked up and they're waiting to get split apart, right? But while they're hooked up, they bind to each other a little bit. And when they bind to each other, they now have a chance to form a swap, to swap a little bit. And this is going to also increase our genetic recombination, because we have here like, I think this diagram is a little hard to understand though. When this picture is good if you can see it, that you see that the blue leg of the left one ends up on the pink chromosome, and the pink leg of the pink chromosome ends up on the blue chromosome. And then once they get split apart, they've become different chromosomes and going to different gametes. So this gives, oh yeah, right here. This gives us recombinant chromatids and recombinant chromosomes. So just to clarify, chromatids are the little legs, right? And here they've labeled these sections as A, B, C, and the pink ones as the lowercase a, b, c. I'll just call it blue ABC and pink ABC, or red ABC. Once we have this little crossing over event, right? This is called crossing over. One of the sister chromatids from each chromosome will have the chance to exchange genetic material with the sister chromatid from the homologous chromosome, called crossing over. And so this formed, this over here will be a synaptonemal complex, and that will allow this little swap to happen. Now we have some brand new chromosomes. And when we have meiosis, we'll have totally, because it's happening all the time, right? You're not passing a one-to-one copy of, you're not getting, oh, I got my one-to-one my father's chromosome set. No, it's swapped a lot with whatever your mom's genes were. So that's how we get lots of cool genetic diversity. And the where was I keep losing my place. No, I wasn't here. We were talking about sex linked, and then, oh yeah, I was here. I just went up. Oh yeah, I was here. Okay. So genetic linkage is basically saying, so that's all to say that there are some genes and chromosomes that swap like that, but there are other genes that can't swap like that. For example, genes that are very close near each other have a low chance of getting split up, right? Because the crossing over would need to happen exactly over here. Oh, that's white. So let's just think of this in a common sense way, right? Which between the red and the green, this thing can get swapped over, right? Anytime, right? Or this part, or this, why can't I draw? Yeah. Or this part can get swapped over at any time, right? If that happens, then the red and the green genes would be unlinked from each other, right? Because they're kind of free to swap, and it's probable that they can swap. However, the blue and the red gene are not probable to be unlinked because they're very, very nearby. So it would have to be a very precise swap right here, right? Like this end of the chromosome would have to go to a different chromosome. And that is unlikely because the blue one is right here. So it's more likely that they would just, go together, right? It's more likely that it's very unlikely like that either we could have, basically it's just a matter of probability, right? Like where will the knife land and cut this, right? If it cuts it anywhere, if the knife lands, wow, this is really giving me a hard time. Let me put this here. If the knife to recombine lands up here, they're still linked. If the knife lands over here, they're still linked. Only can the knife land in this one exact spot to unlink it. This is highly unlikely. That's basically the point. So basically, the closer that genes are, we consider them linked. And we can actually measure this. Traits that are clustered are more likely to be inherited together and pose an exception to the law of independent assortment. Map units or centimorgans are a unit used to quantify the distance between two genes on the same chromosome, related to the crossover frequency, the frequency at which two genes are separately inherited through gene mapping. So I think I have something about that. No, maybe with evolution. No, I don't think so. Nope, never mind. I thought I had something, but I don't think so. I remember reading about it somewhere, but it may not have been this. Okay, anywho. Yeah. Extranuclear inheritance, genetic information that exists inside the nucleus. Oh yeah. So I'm sure you guys know that mitochondria has like its own DNA and stuff like that, and then those are fragments that can go. I wonder if that's a thing. I remember actually I think on my MCAT I had something asking about mitochondrial uh DNA. Yeah, inheritance of mitochondrial DNA. This is such like a small edge thing. I have no idea why they include this. It's really interesting. So I mean, I guess I'll just talk about it. Um, yeah, and Asma, I think this kind of links to your question about like, mutations that get passed on through how do mutations get passed on, right? So the egg actually provides all of the parts of the zygote that the kid will have, right? Which includes mitochondrial inheritance. So you are not getting the mitochondria from your dad at all. You only get them from your mom because your mom's egg provides the parts of the zygote, the cellular machinery. So in that case, the mitochondrial DNA is coming from whatever your mom's mitochondrial DNA is. So it's pretty interesting. Um, the mitochondria is from the mother only. Yeah. So it's just a really interesting like little edge thing. Um, Anywho, so extranuclear inheritance, that's what they were talking about. Extranuclear meaning that outside of the nucleus inheritance of genetic material. And one option for that is the mitochondria. All right. Okay. Now we are on to evolution. So before we move on to evolution, any questions about everything that we covered with our, with genetics and with genes and alleles and all that good stuff? Mm, what was I looking for? Oh yeah, we're about to talk about evolution. Hardy Weinberg, yeah, we're about to talk about this. Yeah. Okay. All right. No questions? No worries. Good. Let's keep it moving. So, I'm sure everybody knows a little bit about evolution from biology, and basically, it's how nature selects for advantageous adaptations to the world, right? And I remember back in was it middle school or something? We did a really nice experiment where the teacher gave everybody like either a spoon or a fork, and then you had to go like take water, like drink water, right? We were all like pretending to be like birds, and we all had to go drink water, and we put our water into our cup. So obviously the people with the forks couldn't, and I was one of those unlucky few who had a fork. If you had a fork, you couldn't scoop up the water properly, right? Unlike the people with the spoon. So basically, if you didn't have like, 15 milliliters of water by the end of 2 days, then you died, right? And that was basically to show us like how disadvantageous mutations or traits will get selected against by the environment, right? If you have something that is interfering with your ability to survive, then you as a trait will be weeded out of the population. So in a nutshell, that's natural selection. And so what I just talked about in my example is the selective pressure, right? I need to get food. I need to get the water, right? But I can't because the more adapted people will out reproduce and out, compete me. And this ties into genetics because the ones with the best traits will have the most success in the environment, and then they'll breed and they'll pass on their genes, and their genes will become widespread and take over, because they'll keep outcompeting the unsuitable genes, so to speak. So let's just go. So evolution has a lot of like just like different traits and concepts to like cover. So I think we'll just go through them like a little bit at a time and see how it goes. So let's see. No, no. Yeah, I think we can just start where up here somewhere. Yeah, okay. So we talked about this. We talked about linked genes. Oh yeah, this is what I was looking for. I wanted to show you guys that there's a really nice trick where you don't need to do the dihybrid cross, right? The dihybrid cross is when you do two alleles, right? Two alleles, and you try to compare. But instead, you can simply do one cross at a time and then multiply the chance. So, what I mean is that if you did this all out, right, and you counted the boxes, you would see that there's like 1 out of 16 or whatever, right? 1 out of the 16 boxes. But if you just do technically this whole giant dihybrid cross is just these things multiplied. I think it's like matrix multiplication or something like that. But if you just do them separately and then multiply the individual things that you care about, you get the chance that you want. So for basically, this person did the D trait and the T trait, and then they wanted to know, okay, what's the chance of the kid being DD capital and TT capital? So if it's a 1/4 chance to have DD capital from just the Ds, and if it's a 1/4 chance to have TT capital from just the T's, then the chance of getting both is this times this. So this 1/4 times this 1/4 gives you 1 out of 16. And this is just a math rule. In genetic probability, when you multiply something, that's the probability of both. And then there's this rule for like the probability of either. I don't think I ever used this, but it is important to know that like when you multiply two probabilities, you get the possibility of both things happening at the same time. Yeah. And so remember we talked about the 3-to-1 ratio and the 1-to-2-to-1 ratio for just a small cross. In this case, for the dihybrid cross, when you cross a heterozygous and a heterozygous, you get a 9-to-3 to 3-to-1 ratio, meaning you'll see 9 different, you can see from here how they color coded the box. You have 9 dominants, because the reason you have 9 dominant is because it can either be capital or it can be capital lowercase, right? So that in that case, you have 9 dominant. 3 dominant recessive, and then 3 recessive dominant, and then 1 recessive recessive. Does that follow? When we okay, so yeah. So this is just a common ratio to know. And like obviously it's better to just like remember, oh, this looks like this rather than like, have to do this out on a test or something. Yeah, we talk about linkage and linked genes and stuff like that. Yeah. Okay. So let's move on to our evolution. Evolution. So natural selection. All right. So we're talking about natural selection. I don't really like how they organize this, so I'm just gonna do my own thing. But you can definitely read this later if you need to. Actually, I encourage you to read it later. Okay, so I'm just gonna go in the order that I think makes sense. So natural selection is, as we said, like for some reason, there's some kind of pressure in the environment that is affecting the population, right? So predators are an option, right? If a predator is able to see blue birds a lot better than red birds, then the blue birds are going to get killed off, right? And the red birds are going to be selected for. So that's what we call directional selection, because the selecting effect is pushing the population in one specific direction. So let's go with this butterfly example, right? Let's say that the predator is really able to see these bright butterflies, but not able to see the dark butterflies because they blend in with the trees or the leaves, right? In that case, the predators are eating up all the light butterflies. And so over time, the population is trending towards being mostly made up of dark butterflies, right? Just to give you an idea of what that means, right? If we have 5 butterflies and this one's white, this one's dark, this one's dark, this one's white, and this one's white, right? And all the white butterflies, or no, not all of them, but let's say most of the white butterflies get killed, right? Then what are we left with? Now we're left, before it was the white butterflies that were in the majority. Now it's the dark butterflies that are in the majority. And when they reproduce, we get something like this, right? And now we have more predators feasting on the white butterflies. So then what happens? Right? And then we have another round of replication or breeding, and then we have it's just getting worse and worse until eventually the white butterflies are going to go extinct, right? So this is what we call directional selection. This population is getting directed in to one extreme. Then we have disruptive selection. Disruptive selection is kind of interesting. Disruptive selection is basically like, we don't like fence sitters. We don't like people who are in the middle. We want it to be extreme. So in a real environment, right? Let's say the predator cannot see the butterfly mixing in with the leaves, so he can't see them, right? And maybe the predator also can't see this really bright butterfly because it kind of blends in with the sunlight or something, right? So then the one that the predator can see is the middle color one, because he doesn't blend in well with the leaves on the ground, and he also doesn't blend in well with the sunlight. So he just kind of sticks out like a sore thumb. Then the predator goes for this one, and what happens is more of them become bright, adapt to the sunlight and become bright, or they become darker and they adapt to the leaves and whatnot, right? So this is what we're saying, that this is called a disruptive selection. We have cut down the middle population, and we are increasing the extreme populations. So yes. And then lastly, we have a stabilizing selection, which is the opposite of the disruptive selection. The stabilizing selection is getting rid of our extreme phenotypes, right? Maybe this one is too bright and it's seen too easily. This one is too dark and it stands out, against the sunlight. So the middle one ends up being the best one for the environment, and that is the one that survives the predator effect. So these are the 3 modes of natural selection. Now, there's also some other. Oh yeah, wait, there's a perfect example right here. I should really scroll down more. So natural selection, right? Frogs get eaten by predators. Notice the ones that blend in better live longer, and now they reproduce more. So now we started with equal frogs. Now we have more green frogs, right? And what else can we talk about? I don't think speciation is just yet. Artificial selection is just like, the humans are the ones causing the selection rather than the environment. But natural selection is just, a catch-all term. Sexual selection is that they have a certain trait that makes their genes more likely to be passed on. So for example, like peacocks do like their, they fan their feathers and whatnot, right? So rituals like that if an animal is better at doing a ritual, right, then his genes is more likely to get passed on, even if his genes are like not necessarily the best for the environment. Kin selection. This is really interesting. This is related to like altruism and stuff like that. An animal. So here's an interesting thing, right? Let's say a monkey is with a group of monkeys, right? And the monkey sees that a lion is on the prowl and it wants to eat them, right? And the lion is going to ambush them. If the monkey screams out, the monkey will get killed for sure, right? And he won't be able to pass on his genes. Is this something that evolution would select for? Would evolution encourage that monkeys scream out when they see lions and they'll get eaten? Actually, this might be a hard question to ask. Basically, what I'm trying to say is it's a really interesting thing that we see altruism in nature. Normally, you would think like, I'm a monkey. I want to survive. I see a lion. I stay quiet and I survive, right? Woohoo, this is a good thing for me, right? Because I get to pass on my genes. However, the downside is my group of animals got slaughtered. Okay? So this is one scenario. Now we make another scenario. I'm a monkey. I want to survive. I see a lion. I scream out and warn the others. I die. My group of animals survived. Of these 2 scenarios, what do you think is more evolutionarily advantageous for the monkey? Self-sacrifice. Why is self-sacrifice better evolutionarily? Bingo. More monkeys survive, right? So it's really interesting. Evolution, it's kind of callous, right? Evolution doesn't really care about as individuals, our survival. but it does care about the species survival. So it encourages this behavior. It encourages this, almost like you can think of it as like a suicidal behavior, right? And actually we see this in biology too, right? If you think about your body and you think about your cells, right? Your cells commit suicide if they realize that they are going out of control and they could harm the other cells and the rest of the body. So this is a very consistent theme that an individual of the collective can say, okay, I can I can die, but everybody else will survive. And so this is something that evolution selects for, and it likes it as a trait. So yes, so in evolution, the second one happens, as you guys said. Because more monkeys will survive, we do have altruism built in as an evolutionary advantage. And what I mean by altruism is I gave up my life so that others could survive. So that's pretty I think very interesting, like psychologically that occurs. Um, okay, so and then I think we're almost done with natural selection, and then we have adaptive radiation. Adaptive radiation is basically like um, these birds are all on the same island, but or they're all very similar, but because so for example, based on the kind of food that they eat, their beaks have changed, right? They all started from the same bird, and they're all pretty similar to that bird, but their beaks have changed because it allows them to have better access to the food, right? And this happens over a long time and a lot of generations, not like right away or anything like that. Um, and eventually, this might lead to speciation. What is speciation? So, what is the difference between, what is a species exactly, right? Like, what separates us from monkeys? What separates us from lions and stuff, right? The difference is that species cannot reproduce. They are reproductively isolated populations. If they cannot reproduce with each other and create a viable offspring, then they are separate species. And you can start from a common species and just by inbreeding constantly lead to totally unique and separate species. And so that's what they were saying, that this can eventually lead to different species being created. Okay. So any questions about natural selection? I feel like that was a pretty good uh, review. And genetic drift. I think genetic drift is what we can talk about next. So again, we're still talking about, the idea of the species and evolution and stuff. So in genetic drift, we have a shift of the allele distribution in an isolated population because of random sampling. So what does that mean? By random sampling, it means, so before with natural selection, the selecting was not random. It was a predator, preying on very, like, this effect is not random. The owl is clearly going for the red toads because they're easier to see. There is a rhyme and a reason, right? However, genetic drift is due to a random event that just randomly kills off a bunch of animals, right? It's not that there was a reason. So, for example, if there was like an earthquake or something and it murdered like half the population, and just randomly it killed off all the white toads, that wasn't the white toads' fault. The white toads didn't die of the earthquake because they were white, right? It wasn't a predator situation. They just randomly lost, the evolution race. And these guys remain. And so what happened here is a genetic drift in the overall population, right? Originally, we had all this variety, and now we have that less variety because of randomness, not because of natural selection. So, um, yeah. Oh, and this kind of links back to, what we talk about with sex-linked sex-linked chromosomes, because the Y chromosome is smaller than the X chromosome. So because it's smaller, it doesn't have as much space to like hold on to alleles and stuff. And so, if there are random mutations or random losses, they probably happen in the Y chromosome. Okay. And the bottleneck effect is very closely related. Basically, it's the idea that the reason it's called the bottleneck effect is kind of fun. Do, 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 do. Yeah, this is exactly the picture I was looking for. So basically, you have a bottle full of like all different types, but when you only take a couple of them out of the bottle and that bottle fills up, then it won't reflect the diversity that was originally there, because only a few can survive through whatever the effect was. So it was a random culling and there was no natural selection for genes. It wasn't related to genes at all. It was just a destruction of most of the population, leading to decreased diversity. How is this different from founder effect? Bottleneck effect is like crushes the population that already existed, whereas a founder effect is like the like because of geography, um, one, I think like a small group of animals leads to that. Hold on. I'm trying to, if there's a picture, it's easier to explain. Yes. So the population leaves, right? And then we lost the diversity. So that's the main difference. The main difference is between, they're both genetic drift. Where when we talk about genetic drift, we're saying that we lose this genetic diversity. From the original population, we go to a more homogeneous and less diverse genetic pool. Why does it happen? The why is telling you what it is. If a small group left, then that's founder effect. If there was an earthquake that killed off half the population, that is bottleneck effect. Does that make sense? Okay, good. So we were looking at bottleneck. Okay. Well, no. Bottleneck. Okay. Um, differential reproduction. Yeah, we already talked about this basically. Like, whoever survives is going to have the best chance at, passing on their genes. So that's just, it's differential reproduction because the reproduction of the whole species is, trending towards the best genes. We talked about group selection already with the monkeys, right? We're not always selecting just for an individual, we're selecting for a group. Okay, inbreeding. Everybody knows inbreeding is bad, but why is inbreeding bad? Inbreeding is bad because, we've been talking about genes this whole time. We've been talking about those recessive traits that can be masked by healthy dominant traits. But if you keep inbreeding, then you will increase your chance of those recessive traits popping up. So the more recessive traits that you have, the more you're going to have a bad time, basically. Um, and yeah, there's a reason that genetic diversity is very, very important, and we have so many mechanisms to increase our genetic diversity, because genetic diversity is the way that we acquire new adaptations that can help us survive in our environment. But if we're constantly inbreeding, we're never giving ourselves a chance to get new traits. We're only like locking ourselves into old and recessive traits. So this is not good for survival. Okay, so we had briefly mentioned about speciation. We had briefly mentioned reproductive isolation. So reproductive isolation is the idea that that they can no longer mate with each other, so they are totally different species. So what does that mean? How does that happen? Okay, so there are two different kinds of reproductive isolation: pre-zygotic and post-zygotic. So pre-zygotic means that they are reproductively isolated because they don't even get a chance to mate and have a zygote, right? It's not that they're trying, it's that they can't even try, right? So why would that be the case? Well, the first one would be maybe ecological. Maybe they live in completely different habitats and they never even see each other, right? Temporal. They could live at different times or different seasons or whatever, right? Like a nocturnal animal is not going to have any interaction with a strictly what's the opposite of nocturnal? I don't know.
Diurnal.
What is it?
Diurnal.
Diurnal? Oh, cool. Diurnal. Day. I guess that makes sense. So a nocturnal animal wouldn't have any interaction with a strictly diurnal animal. Then we have behavioral. This is just like, a catch-all, like, if they don't behave the same way, then they're not gonna be able to I think this is mainly referring to like mating rituals and stuff. Like if they don't respond to the same rituals. Mechanical is obvious if they don't have compatible genetic or genital organs. Gametic is that, okay, let's say all of this is working, but when it comes down to it, their sperm and their eggs are not compatible. So then, no good. So those are all the different possibilities for why species would be isolated from each other. Now, what if none of these are the problem? Like they actually can mate and they can have a zygote, right? Now, they're not in the clear yet. There can still be some issues, right? So for example, the first one is called hybrid inviability, which is to say that the hybrid offspring is born but dies. It cannot survive, right? So hybrids are produced but fail to develop to reproductive maturity. So this is, remember, we're talking about the species. We're talking about passing on genes, right? Just because this animal was born doesn't mean that it is, it is capable of passing on the genes, right? Then we have hybrid infertility. This is an even better example because we have an animal, right? You might know that a mule is the offspring of a horse and a donkey. However, a mule is born sterile. Because it's sterile, it cannot pass on its genes. So this is, it is a reproductively isolated species. And lastly, like we could go even further. Maybe, for example, the mule type animal could give birth, but those guys don't have the ability to reproduce, right? So that's called hybrid breakdown. It was working, but then it broke down at the second generation. So these are all different ways that animals can become reproductively isolated and parts of different species. Yes. Okay, this is a fun one. All right, so this is cool. So now we'll talk about speciation. So speciation, let me see here. Speciation is basically, like we said, two different populations of organisms are considered different species when they can no longer interbreed and produce viable offspring, right? And so now, how does this happen? How does this happen? Where am I looking at? Anagenesis, cladogenesis. I guess I don't have a picture of this, but these are just two different types of speciation. Allopatric speciation is because the two populations were geographically separated and became genetically distinct. This could be like with founder effect, but it's more like there's just two, they were together, but then they got split up, and then split up for so long that by the time they, reinteract, they are no longer able to mate with each other. And sympatric speciation is that they are in the same environment, but they have adapted differently. So they adapted, like we were saying, nocturnal or diurnal, or maybe their mating rituals and stuff don't align anymore. Those are basically two different ways that they can become speciated. Now we have this idea of anagenesis and cladogenesis, right? And I believe genesis is birth. So it's like, how was the species born? Anagenesis is that the species we're getting reproductive isolation without any splitting, right? It's just the next generation cannot reproduce with the older ones and so on and so forth. This is not that common. More common is that we have cladogenesis, which is splitting, right? And so the splitting, because this guy, so for example, right, let's think about how could this happen? Maybe it happened through sympatric speciation. Maybe this purple dinosaur, right? Maybe some of them, were decided like they could, I don't know how to say this. Maybe some of them went off to like a different environment because there was more food there or whatever, and some of them remained, right? And so the ones that remained became blue, and the ones that went became brown. And because they spent so much time apart from each other, they lost their, their mating rituals changed and their behaviors changed. And then if you reintroduce them to each other, they would no longer be able to mate. They would be different species. And of course, we're talking about, especially with dinosaurs, we're talking about the order of like, thousands of years, right? Because it's, well, maybe not thousands, maybe hundreds of years. But basically, we this process is much faster for animals that have much more faster generations, like fruit flies and bacteria and stuff that grows like much faster. But for mammals, it takes a minute. Or actually, not mammals, reptiles, right? Dinosaurs are reptiles, right? I think so. Don't quote me on that. Anyway, structures. All right. So structures. Oh, I don't have a picture. No, I can find one. Yeah, okay. So homologous structures are physical features shared by two different species because of a common ancestor, right? So these are homologous structures. And we have basically it's a conserved motif in our anatomy, in how we all have very similar bone structure in our arms. That's what they're saying is a homologous structure. Now, the opposite would be an analogous structure, which is not conserved. Like this. A bird wing and an insect wing are completely different structurally, but serve the same function, right? So what does this tell us? It tells us that these guys did not evolve from the same ancestor and does not have the same evolutionary lineage, right? Just because they do the same thing, help with flying, doesn't mean that they evolve the same way. So these are analogous structures. Homologous, homo meaning same, from the same origin, common ancestry. Analogous, similar, but not coming from the same origin. Okay, good. And now this last main thing that we have to talk about here is the different kinds of evolution. So we have divergent, parallel, and convergent evolution. In divergent evolution, this is what is cladogenesis, as we saw here, because this purple one diverged into two different species. So in divergent evolution, we start, with one species and it diversifies into two species, right? Here we have two A's, like you can think of it as like two different populations of the same species. One population of A became S, one population of A became T. Okay. Parallel evolution is when there are two different populations, but because they're experiencing the same thing and they have the same responses, they both evolve into similar traits. So for example, if we looked at here, if you looked at these birds, right? Notice how the bird that digs for grubs and the bird that digs for insects have very similar beaks, right? Because they're both doing it for the same kind of thing. One is digging insects, one is digging for grubs, right? So these guys can, if they are very close, can be considered to be having this kind of parallel evolution because they are evolving away from the founder species with this pointy beak, but they're evolving in a very similar way because the pressure, the food pressure that they have in that environment is similar. So that's basically what we're looking at as parallel evolution. And then convergent evolution is when two different species end up having very like analogous structures, right? So we saw like birds and bugs can both fly. They both have wings, right? So they converged on that ability to have wings, but they're totally anatomically and evolutionarily in terms of origin, they're different. Okay, so I think there's not that much. Yeah, I don't think there's that much left here. I think the one major thing to do is cover Hardy-Weinberg. Then we'll see. Hmm, symbiosis is, I don't remember if this ever came up, but it's just some definitions you could know. Commensalism is that one species is benefiting. So this is about, so symbiosis is like, how species interact with each other. So you might know about like, parasites that they go in and they hijack a species, right? If they hijack the species, the other species gets hurt. Mutualism is like coral and like clown fish, or what are they called? Whatever the fish that Nemo is, right? Both species are benefiting, right? They have a good effect on each other. Commensalism is less common. One species is benefiting while the other one isn't getting anything or being hurt by it either way. So okay, so the last thing to do is talk about Hardy-Weinberg, but let's just see if there's anything that we've missed here. Talked about genetic drift, genetic leakage. Do we have genetic leakage? No. What's genetic leakage? Occurs as genes are transferred between species through interspecies breeding. Oh, this is relevant to what we talked about with like hybrid breakdown and stuff. But yeah, here we have the bottleneck effect and the colony, the founder effect, right? And okay, and now we can talk about Hardy-Weinberg. The molecular clock, theoretical method of dating the evolutionary age of a species based on the frequency of allele changes. The higher the degree of genetic difference between the two species, the longer ago they were assumed to have differed. Oh, okay. So basically they're just this is related to like taxonomy and stuff, like no, this isn't a good. I can't find a good picture, but basically like, you know, the less genetic diversity that you have between a species, the more recent of an ancestor they are, right? So we're supposed to have like 99% DNA match with like monkeys and stuff, right? Or was it orangutans or something? So basically they're supposed to be we based on the degree of genetic differences, we can tell like who's similar and who's dissimilar to us. Okay, so I think this is probably going to be the last thing. Okay, any questions about, so we basically just covered the entirety of evolution. So any questions about evolution in general? And all that stuff. It's really not that hard. They don't ask, it's really just a matter of like knowing the definitions. Okay, so let's go to Hardy-Weinberg. Hardy-Weinberg is tricky. Hardy-Weinberg is the only time that math shows up. Well, one of the few times that math shows up in biology. So you just need to know how to do it so you don't get tripped up by it. Hardy-Weinberg equilibrium is essentially that there's it's a way for them to kind of describe a population, to describe the changes of allele frequency in a population, right? Hardy-Weinberg is pretty much like totally theoretical. Like it's not really, it has, I don't want to be mean, but it basically doesn't have any like real world um usefulness at all. But basically, the Hardy-Weinberg equilibrium is the idea, it's describing a scenario where a population is not evolving. And for a population to not be evolving, it would be that there's no there's no sexual selection or preference in how the individuals are mating. It's purely random, right? Then all phenotypes have to have equal viability and reproductive success. So there's no natural selection. The population is infinitely large, so there's no, small population pressures. There's no gene flow or migration, and there's no genetic drift. So this is all like completely theoretical. And basically these are all in place so that there is no population evolution, right? So now we get to the math. Okay, so we have two sets of equations that are describing the traits. We have P and we have Q. P refers to the dominant allele. Okay, so now if we have a dominant trait and we have a recessive trait, then in each individual of the population, right, we can get three different genotypes. We can get PP, we can get PQ, or we can get QQ. Now, we have noticed that we have two PQ here rather than just PQ. Why do we have two PQ here? Right, the Punnett square, exactly, right? Because we get P and P here, then we get P, Q here, get P, Q here, then we'll get Q, Q here. So that's how we get our 2PQ, right? So these are our, this will account for our whole population. So now, because this accounts for our whole population, we get this equation. These genotypes are equal to 1, and also the individual alleles equal 1. So now, this allows us to estimate the allele frequency as long as you know one genotype. So if they give you like, this is the dominant allele and this is the recessive allele, then you can plug that in and you know how much of the population is going to be homozygous dominant, because the amount of homozygous dominant is right here, this PP, right? So PP is P 0.4 times P 0.4 is 0.16. So then 16% of a population is homozygous dominant. Then if you're supposed to know 2PQ, 2 times 0.4 times 0.6, 48% of the population is heterozygous. Q, you do the math, 36% of the population is homozygous recessive. And if you add up all of these percentages, you get 1 or 100%. And that's how Punnett squares work. So, I mean, that's how the Hardy-Weinberg equation works. It's not the worst, but it is a little annoying if you forget how it works. Yeah, so I think that is about it and good. So we have a little bit of time so for us to do some practice problems. Any questions right now? Does it make sense? Okay, let me see, where were we? So, does this have any problems? It does have some problems. Oh, interesting. Ooh. What is this? SNP mutations. Oh, kind of cool. Okay, so here's what, do you guys want to do questions now? Okay, so here's what we'll do. I will leave this on the screen. Actually, let's take what time is it now? 12:25. Let's take a quick 5-minute break. And then you can look at the question if you want. Let's take a quick 5-minute break and then we'll come back at 12:30 and we'll start working through this. I'm going to post this here. The question is at the bottom of the thing. You can look at that and we'll be back at 12:30. Okay, so let's go through this passage. Okay. Do you want me to read it? Or did everybody read it already? Okay, I'll just read it. Let's find out because I gotta know what this is about too. Human societies living in highlands and mountainous areas encounter many inhospitable factors within their environment. Ancient and modern societies living in central Andes above 2,500 meters above sea level are directly affected by low oxygen pressure, which may lead to hypobaric hypoxia and cold temperatures. Descendants of ancient Andean populations display traits that include elevated concentration of hemoglobin, thoracic expansion, increased lung capacity, and higher levels of pulmonary arterial pressure. These traits are theorized to ameliorate the negative effects of hypoxia that may be encountered at high elevations. So far, this hasn't given me anything useful, really. These traits are considered to have arisen through one of two possible paths. One in which exposure to low oxygen levels promoted adaptation through natural selection, and one in which a previously existing phenotype enabled humans to live and thrive in an otherwise hypoxic environment. 3 different single nucleotide polymorphisms in 2 different candidate genes, okay, were identified as target for analysis, right? So here, this is what we talked about with the point mutation, single nucleotide polymorphism, okay? So we had a point mutation, and then notice here, they said 2 different candidate genes. So what are the candidate genes? We have NOS3 and EGLN1, right? These are our candidate genes. And then they said, what is the polymorphism? They're saying at this position, a guanine was replaced with a thymine, or a thymine was replaced with a guanine, or an adenine was replaced with a thymine. These are the 3 different polymorphisms that they're investigating as potentially something that either promoted adaptation for for survival with low oxygen. That's what they're researching. So they analyzed paleogenomic data from 150 prehistoric individuals who lived between 8,500 to 600 years before the present year. These individuals were further identified to have lived along the ancient coast, a low altitude environment, or in the highlands, a high altitude environment. PCR and base extension assays were used to genotype these 3 altitude adaptation associated SNPs. Okay. So I have a table here, and the table is showing us what? So it's showing us the prevalence, or how much of G was there versus T, right? And we have to think about, which one are they saying is the one that supports surviving in this hypoxic environment, in this low oxygen environment. So on the coast and the highlands, right away, we need to turn that into, the concepts that they are talking about. They're talking about oxygenation, right? Hypoxic, non-hypoxic, whatever. So we need to say, they told us that the coast is a low altitude environment. So if it's coast, if it's low altitude, that means there's high oxygen for the ancient coast. And then in the highlands, that's high altitude, literally called the highlands, altitude, and it's low O2, right? Just so we don't get confused, we make it simple for ourselves. Then we see that we see a trend, right? Age in generations, T mod minus N. What is T mod? T mod. Oh, maybe modern? Time modern minus N? Time modern. So whatever it is now, minus this many years. So the oldest generations, so these are the oldest generations. So it's oldest to current. Okay, so that's basically what it is. So we see that the older generations on the highlands, the oldest generations have 75% G, and then the closer we get to now, the more G that they have, right? So we see that the amount of G is increasing the closer we get to the current times. So this is suggesting that G increasing is doing what? the more G, that G, as G increases, what's happening for the Highlanders? Well, actually, so T is decreasing, right? T is decreasing. T mod minus N. This T mod minus N is throwing me off. Age decrease in generations. So is it like, is this saying that like, I'm trying to figure out, is the 22 the oldest or the youngest? It's the youngest, right? It's like the most recent generation. I think that makes sense. So then, okay. Anyway, hold on. Let's not get confused. So what I want to do is look at the questions first. So they're saying the S&P mutations investigated in the study were most likely the result of the bottleneck effect, the founder's effect, the genetic drift, and the migration, right? So we have seen all of these four concepts today. And basically, we should be able to say, okay, this is this because this, and this is not this because not this, right? So they're saying S&P mutations. They're simply talking about the mutations right now. Why did the mutations happen, right? And what did we know about? We know that the big thing is that there's different places, right? They're different places. There's the ancient coast, which is low altitude, and the ancient highlands, which is high altitude, right? So first of all, is it going to be the bottleneck effect, right? What is the bottleneck effect all about? In one word, right? Like, what is it mostly related to? Good. Disasters. Okay, low diversity, right? Here's the problem with low diversity. All of these things can relate to low diversity. So because the founder, yes, so the founder effect is also low diversity. The genetic drift is also low diversity. Migration could lead to low diversity. So we're looking for a very specific word that's going to allow us to decide that this is right or this is wrong, right? We want to know how these things are different, right? So maybe like the most characteristic word, disasters. Disasters is a good word because it tells us exactly what the bottleneck effect it is all about, right? Disasters that leads to low diversity, right? Everything here is leading to low diversity. What about the founder effect? What does the founder effect do? How does it lead us to low diversity? A small group, right? Yeah. So a small group branches off, and that gives us low diversity. Oh my God, this is a nightmare. Small group, low diversity. Okay. How about genetic drift? What about genetic drift? Random, exactly, right? Random stuff that's happening, just a random changes that a population can go through when they are not being directed by selective pressures, right? So random leading to not necessarily low diversity, I guess. Like, I guess low diversity isn't really correct here. It's just random changes leading to different things. Migration can definitely lead to low diversity, similar to the founder effect. Okay, so in this case, right, the mutations investigated, right? Did they mention anything about a disaster? No. Did they mention anything about Small groups? I don't think so. So then we can think about if it's genetic drift or if it's migration. Did they say that they migrated to the highlands or anything like that? Living in highlands and mountainous areas encounter many inhospitable factors with their environment. Descendants of populations. But it would be founder or migration if they are talking about, if they said like some people went up to there from the original group, but I don't remember if they had said something like that. Human societies living in highlands and mountains areas encounter many hundreds. Ancient and modern societies living in central island days directly affected by low oxygen pressure. There are two different regions. But for it to be founder effect, one group has to go to the other region from one to another region. Yeah. So in that case, I think it might just be genetic drift because they kind of ended up there, and then because they were there in that environment, they had these small changes that were over time slowly helping them to adapt to that environment more. Trace or theorized to ameliorate the negative effects of hypoxia. There's two reasons I don't think it's founder. The first is that they never explicitly say that anybody went there. And the other thing is that because founder and migration are so similar, I already don't like these as choices, because then I have to make a choice between founder and migration, and I feel like they're too similar for either of them to be like a good choice. That's like a meta reason, not relevant to the question. How is it random? Because these single nucleotide polymorphisms are just random mutations, right? Mutations are just random. Are bottleneck and founder effect under genetic drift? Yes, they are under genetic drift. Well, they're types of genetic drift because there is randomness in how they're more specific types of genetic drift. Let's say it that way, right? So founder effect and bottleneck effect are types of genetic drift, but not all genetic drift will be bottleneck effect or founder's effect, if that makes sense. Yeah. So it's a little tricky because there is a focus on regions here, right? But the thing about it is that the founder's effect is very specifically, there's always a regional influence, right? Because there's always the environmental influence of your region, right? What makes it a founding effect? If a population split off to go to a brand new region, right? The population that's still there still experiences the effect of the region that they're in, right? That's not a founder effect. They're just there. So in this case, I feel like they're leaning more towards that like they're in this region, there happens to be low oxygen, they happen to have these random small polymorphisms that may be helping them with that. Yeah, so that's hopefully correct. Do you want to check the answer now or at the end? Later. Okay, good. So let's go question 2. What is the expected number of individuals heterozygous for the RS1 blah 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 813 mutation in the population of 1,000 individuals living? Okay, so this is a Hardy-Weinberg. Okay, So when you see expected number of individuals heterozygous, this is hardy one. Okay. So now what we need to do is go back to the RS, this mutation, right? And we have to see what it looks like. 813. 813. Here's the 813, right? And here's what they gave us. They said, and where did they say? Among the ancient coast. Okay, so among the ancient coast, wait, did they give us a time? What is the expected number of individuals heterozygous for the mutation in a population of 1,000 individuals living among the ancient coast? Oh, I see what they did. They're all basically the same. There's no change here. So you can use any of these. I was looking for a time because I was like, what, like which data set should I use? But they're all the same. They're all 0.4 and 0.6. So we will use 0.4 and 0.6. And then they said for the mutation in a population of 1,000 individuals. So it doesn't matter what I select as my P or my Q here, because I want the heterozygous anyway. So 0.4 and 0.6. So let's write this out. So my first equation, P plus Q equals 1, right? So P is my 0.4 plus my 0.6 equals 1. Okay, that's good. Now P squared plus 2PQ plus squared. It's going to give me 1,000. That's my population, right? So now, hold on, I haven't done this in a while, so I'm just going to try this and see if it works. We'll see. P should be 4.4 squared, 0.4 squared, whoops, squared plus 2. Actually, wait, do I even need to do this? Wait a second. 2PQ. Yeah, wait, let me just do it this way. Let's just see, let me just see if this is the right way. Q 0.6 squared equals 1,000. Okay, so 0.4 squared is 0.16 2PQ plus 36 equal to 1,000. Then math. Then what? I think 36 52 plus 0.52 plus 2PQ equals 1,000. So then then the 2PQ is going to be the 1,000 population minus 52. This is not I know this is not properly written because this is not correct, right? Because this is supposed to be like 52% of the whole population. I just don't know how to properly write that. But I know this is going to be minus 52% of the 1,000, right? So of the 1,000. So that's 2PQ equals 1,000 equals, that's 520, right? Okay, so then 2PQ is equal to 480. And that's, so then if I probably screwed this up somehow, but what is the proper way you're supposed to do it? Hold on. Let me see. I'm pretty sure I have a better example somewhere. Yeah. Does this make sense though? I'm just confused why I think I skipped a step or something here that makes this not work. Oh, wait a second. I think I'm not supposed to make it 1,000. I think I am supposed to do that at the end. Okay, yeah. Yes, there we go. Uh-huh, there we go. And so now you have the percentage of the population that is heterozygous, right? So now what is my population? I've got 1,000 individuals times 0.48, and now I know I have 480. 480, that should be correct. Okay. Any questions on this? See, I told you I'm not good at math. So don't that's about the best I can do for math. Okay. So that's how I'm pretty sure that's correct. It should be 400, it should be 480 individuals heterozygous for the mutation in a population of 1,000. All right. Assume that some individuals who inherit these SNPs express increased cardiac vascularization and increased arterial oxygen saturation. Which of the following can be concluded? Increased cardiac vascularization, increased arterial oxygen saturation. So they talked a little bit about it somewhere. Thoracic expansion, increased lung capacity, higher levels of pulmonary arterial pressure. Theorized to ameliorate the negative effects of hypoxia. So it's theorized to deal with hypoxia. Inherent these SNPs express increased cardiac vascularization, increased blood flow, and increased arterial oxygen saturation, which can be included. Individuals in this population have inherited structural abnormalities. I'm not a big fan of the word abnormalities here. I think they would just say mutations. I'm not sure why they said abnormalities. The population is no longer in Hardy-Weinberg equilibrium. Why do they bring up Hardy-Weinberg here? Hardy-Weinberg, the population is no longer in Hardy-Weinberg. Hardy-Weinberg equilibrium means there's no evolution, right? No longer in evolution. No, it's no longer in no evolution. This one's really weird. Hardy-Weinberg means no evolution is happening. So they're saying the population is no longer in no evolution, or the population is evolving, right? This is a double negative because it's no longer in no evolution. So that's basically saying the population is in evolution. Right, that makes sense though. The population, if there's changes happening, then the population is evolving, unless I just confuse myself. I think that's correct. Hold on. Wait, let me just go see what the Hardy-Weinberg thing was. Yeah, the population is not evolving and said to be in Hardy-Weinberg equilibrium. That makes sense, but let's look at the other ones. The trait must be sex-linked recessive. No, they didn't say anything about that at all. No. There's nothing about that. So I can't say either way. The trait must be co-dominant. Okay, interesting. Can the trait be co-dominant? No. You can't be expressing both of these things, right? Can you? I've never seen codominant in this context, but it seems like you can only have a T or an A, right? So if you can only have a T or an A, then you couldn't express codominance. Unless I'm misunderstanding that. I don't know. I don't know this like notation well enough to tell. I think I'm gonna go with B just because it's like a very convoluted choice. So I feel like they were trying to trick me, but this makes sense. The population is no longer in Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium means that there's no evolution. So they're saying the population is in evolution. And that's correct, because if they're having these changes that's leading to these anatomic changes, then that's evolution, right? And those are happening, although those are happening randomly. No, that's okay, because Hardy-Weinberg is supposed to be against randomness. No, it's for, oh no, I'm getting confused. This is a tough one. You're leaning towards A? And the only reason I don't like A is because I don't know what abnormality means. I don't know what abnormality means. I don't know if that makes sense here. It does, I mean, they have structural changes. I don't know if it's an abnormality. I would be so happy to pick A if this just said structural changes or structural something. I don't know if an abnormality is fair. But I'm not sure about this one's a hard one. I don't know. Why would they say abnormalities? That's true. I'm just thinking about in terms of like, would we call it abnormal? Or will we just call it different? Will we call it a mutation? Well, a mutation is sort of an abnormality. I just hate the word. Okay. I don't like the word. I think A is fine. I think A is, I mean, A makes sense. This one's a hard one. I think it's either A or B. I think C and D don't make any sense. There's nothing related to them. It's either A or B. Okay, we'll find out. Anywho, upon additional analysis, researchers discover that the two EGLN mutations are often inherited from each other. Which of the following laws has been shown to be violated? So inherited from each other is saying that if you have this mutation, then you end up getting this mutation. Or if you have this mutation, you end up getting this mutation, right? They end up going the same, right? So which of the following laws has been violated? So which of these laws is saying that mutations are not inherited from each other? Yeah, A seems to be the only one that makes sense. I don't even know what these are. Law of segregation. Wait, no, is it the law of segregation? No, segregation is wait, is it A or C? Segregation, independent assortment. Researchers discovered that the law of segregation is just that you can have separate gametes. So then no, it's A. Yeah, it's A. Because the law of independent assortment gives us that genes should be taken up independently. However, in this case, these mutations are not being taken up independently. They're being inherited from each other. So A is the one that seems the most violated. Okay. Now, suppose that compared to the modern population, all ancient peoples who lived in the highlands displayed higher tolerance to hypobaric hypoxia. However, individuals within the ancient population displayed high amounts of variance to this tolerance. This trait is best described as expressing, oh, okay, penetrance and expressivity. All right. So, all right. So let's work through this one together because we spent good time on penetrance and expressivity, right? So individuals. So suppose that compared to the modern population, all ancient peoples who lived in the highlands displayed a higher tolerance to the hypoxia. Okay. However, individuals within the population displayed high amounts of variance. So when we see high amounts of variance, what part of the answer choice is that talking about? Is it talking about penetrance or expressivity? Good, expressivity. Now, if they're saying high variance, is it broad or narrow expressivity? Broad, right? So immediately we remove B and we remove D, right? So it's only about now between complete and incomplete penetrance. Now, what word in this answer choice, in this question, is going to help us choose between complete and incomplete? Very good. All ancient peoples. It didn't say some, it didn't say most, it said all. This is an all or nothing thing when it comes to penetrance. It's either all or nothing. So it should be A, all complete penetrance and broad expressivity. Okay, so now we can review our answer choices. So what did we, yeah, go ahead. So incomplete, so penetrance was about, so penetrance, if we go back, penetrance, incomplete and complete. Complete is that, and I posted it in the chat earlier. If you scroll up to 1123, we had said that if you have the genotype, if you always express the phenotype, then it's complete penetrance. But if anybody has the genotype but doesn't express the phenotype, then it's already incomplete penetrance. Okay, and so since they said the Highlanders all displayed the higher tolerance, then this is complete penetrance. Nobody didn't express the phenotype. Okay, so let's review. Let's see. So what have we said? We said, I think we said question 1, we said C, then we said B, then we said oh, we couldn't decide for 3. 3 was either A or B. Then we said A, then we said complete penetrance and broad expressivity A. So this is let's see. Okay, so answer choice C is correct. Single nucleotide polymorphisms are mutation at a single DNA nucleotide within the genetic code. These mutations occur randomly and at chance. Okay. Bottleneck and founders explain drastic change in frequency that may be present after a migration event or natural disaster, but does not apply to the onset of genetic mutations. Migration is an event that refers to the movement of individuals across a geographic region. So the main reason that it's not the founder or the it's not bottleneck because they didn't mention disaster. It's not founder because they didn't mention migration, and it's not migration because they didn't mention migration. Ultimately, that's why C is like kind of like a more broad answer, but it's the least wrong answer. B, oh yeah, 2 is B. Yeah, okay, so this is calculated properly. You get the 0.48 and then you multiply the times 1000 and you get 480. Okay, so 3. B is correct. To meet the Hardy-Weinberg conditions, a population cannot experience changes in allele frequency. Thus, the population cannot undergo natural selection. That doesn't make any sense. To meet the Hardy-Weinberg conditions, a population cannot experience changes in allele frequency. Yes. The population cannot undergo selection. Yes. But why is that correct? I know all they did was explain Hardy-Weinberg. The population is no longer in Hardy-Weinberg equilibrium because they're having changes. That's it? A structural abnormality is a change in a localized region of one or more chromosomes. Oh, I've never heard this terminology before. Structural abnormality. Chromosomes. Structural chromosome abnormality. Ah. So this is something, so I never even heard of this before. And also, you see what I'm doing now? Where like, I'm going through a question and I'm like, what the heck is an abnormality, right? I never heard of this. This is something I'm going to take a note of. If I'm studying for the MCAT, I'm taking a note of this and I'm putting it on a flashcard or something, just so, I know what a structural abnormality is now. Oh, it refers to these mutations. Okay, that's kind of stupid, but we know what chromosome mutations are. We've just never heard it referred to as an abnormality, but now we know that there are abnormalities, right? So we know, so the good thing is there was no evidence for any inherited structural abnormalities, so it's not A. B is, I'm annoyed at their explanation here because it's not very good. They're just saying that, oh, okay, I get it. Okay, so let's write out a logic chain. So the logic chain is, we're starting from the fact that we have S&Ps, there are S&Ps, right? So the S&Ps lead to natural selection, right? No, I'm jumping ahead. The S&Ps are leading to increased cardiac vascularization and the oxygen thing. And arterial O2 saturation. Right? This is increased natural selection because these things are helping the Highlanders adapt to their low oxygen environment. If it is natural selection, if natural selection exists, then there is no Hardy-Weinberg. Because Hardy-Weinberg is when there's no evolution or natural selection. And this is, the population is no longer in Hardy-Weinberg equilibrium. So that's basically what they were getting at. But it was very annoying to understand. Does that make sense for 3? Okay. Sure. Yeah, sometimes you'll run into questions where you're like, okay, fine. Like that, all you can do is, take it for what it is. I think we had said A. Yeah, answer choice 4 is A. The genes are found to be linked. Genetic linkage is most often due to the close physical proximity of genes on a chromosome, meaning they are less likely to sort independently. Choice A is correct. Expressivity refers to extent. We talked about expressivity already. Segregation states that alleles are divided into gametes such that each gamete receives an equal number of alleles. Choice C is incorrect. It doesn't have anything to do with this. I'd never even heard of the law of parsimony before. So again, it's not the right or wrong, it's a wrong answer, but I'm going to look at it because you never know when it could be the right answer, right? Law of parsimony, DNA, or whatever it was, or genetics. Hypothesis of relationship that requires the smallest number of character changes. These character changes are DNA. So the smallest number of DNA changes. All right, sure. I mean, I'm sure that's fascinating for whoever came up with it. Now, choice 5. Yeah, I think we got this perfectly, right? Complete penetrance and broad expressivity. A complete penetrance, but everybody understood that one. Okay, so that is it. 1 second.